Welcome back, students. Today we will study the anatomy of monocotyledonous root. In the previous class, we had seen the anatomy of dicotyledonous root. So, just like the dicotyledonous root, our approach will be same. In order to study the monocotyledonous root, we will take a root, cut a thin slice, then after adding the appropriate amount of dyeing substance, we will observe under the compound microscope. So in the ground plan, we will see how the epidermal tissue system, ground tissue system and vascular tissue systems are arranged in a monocot root. So if we see the ground plan and the monocot root, then somewhat similar to the dicot root, but not actually. There is slight variation. If you see the ground plan, though the root hairs, epidermis, cortex region is there, but inside the stellar region, which consists of the endodermis, beyond endodermis, we will see various patches of vascular bundles are found. That's why we call this type of arrangement as polyarch condition. So, the outermost layer is the epidermis, also known as this. Just recall from the last class, we had called it as epiblema. So, the outermost layer is the epidermis. Beyond that epidermis, we will have cortex. But here is the key point that in cortex, we observe it is hardly compactly arranged, but here it is loosely arranged. So here in a monocotyledonous root, what we will see that the cortex region, based on the presence of intercellular spaces or how the cells are arranged, it is been divided into two parts. Or the outer one, which is found attached to the epidermis, or we can call it as outer cortex, and this is compactly arranged and having no intercellular spaces, and next is having intercellular spaces. So this is a parenchymatous cell. But in dicot root, we have seen that intercellular spaces was present right from the epidermal region. But here it is arranged, it is compactly arranged, thick-wide, and it is having a intercellular spaces. But both are of parenchymatous nature. Then importantly, the cuticle is absent here. So both dicot root and monocot root, remember, there is no presence of cuticle. And beyond cortex, or the innermost layer, is the endodermis, and this endodermis, there are Casparian strips. So some of the walls, some of the cells, the walls gets deposited with suberin, and these Casparian strips, they prevent the water entering the xylem. Then how the water gets transported? So other cells in which Casparian cells or the Casparian strips are not found, these cells are known as transfusion cells. And these transfusion cells help the water to be conducted by the xylem or the vascular bundles. Then beyond the endodermis, we will see a single layer pericycle, and the pericycle here also gives rise to only lateral roots but not to vascular cambium, and that's why in monocot roots also there is no secondary growth. Then after pericycle we will see the vascular bundle. So here see, these are the xylem. Just count 1, 2, 3, 4, 5, 6, 7. So it is beyond 6. So that's why it is known as your polyarch. Just remember, it is polyarch condition, and it is the characteristics of your monocotyledonous roots. But in dicots we had seen 2 to 4 patches, but here it is beyond 6. So up to 6 we can find in dicots, but in monocots it is in polyarch conditions. So what is the meaning of polyarch? The vascular bundle or the vascular patches are found beyond the number 6. So that's why it is your polyarch. And here also the arrangement of xylem. The arrangement of xylem is important. The first one or the protoxylem. Just remember, the protoxylem is found towards the periphery, and that's why it is also in the condition which is known as a exact condition. So here in monocot roots also the arrangement of protoxylem is of exact region. And another term that you might be studied or in a tissue system we have seen, if xylem and phloem are present, then what is the term used? Just remember. Then if vascular cambium is present in between xylem and phloem, then it is called open type, but here it is closed type. We have seen no vascular cambium, not in dicots, not here also in monocots. That's why it is a closed type of vascular bundles we are seeing. So it is a polyarch region, polyarch, then in exact condition. Just remember these terms. So based on these terms, we will be expecting questions in your examinations. Then these cells which is found in between the xylem and phloem, they are of parenchymatous nature and they are called as conjunctive tissue. So just remember this value, it is conjunctive tissue. Conjunctive tissue. So what are these conjunctive tissues? The tissues. This is compact layers. This is phloem. This is xylem. So in between xylem and phloem, we have the presence of parenchymatous cell, and this is known as your conjunctive tissue. And here, this portion in dicots we had not seen that, but in monocot we see that the pith is well developed and large enough. See the diagram? The pith is large and well developed, and as pith is present, so pericycle, your vascular tissue or vascular strands, and pith, they together constitute the stele. So stele is present in monocot root as it was present in a dicot root, but in dicot it is very small or inconspicuous. So the anatomy of monocotyledonous root, we see that the epidermis is the outermost layer, and this epidermis or epiblema contains various unicellular root hairs. These root hairs help in the absorption of waters, and epidermis, the next layer of the epidermis is cortex. The cortex we will find two kinds of cells. One is thick wall, another is thin wall. As the walls become thickened, we will find no intercellular spaces, but here intercellular spaces are present. And the innermost layer is endodermis, and beyond endodermis we will say pericycle. It gives rise to lateral roots but not to vascular cambium. That's why secondary growth is absent in monocots roots. And next is pith is well developed. Xylem, phloem are present in more than 6 patches. They are in polyarch condition and in exact nature. That is protoxylem towards the periphery or epidermal region, and metaxylem towards the center or the pith region. So this type of arrangement is exact condition. So let's look a question and how we can upper also write the answer. So like that, features of a, so here features means anatomical features of a monocot root. So starting from a epidermis up to the point of pith, we will write serially. Epidermis, single cell thickness, beards, root hairs, cortex, thick wall and thin wall, that's why they have presence of intercellular spaces. Then endodermis is the enormous lateral cortex. Then the xylem and phloem patches are present in 6 patches, in more than 6 patches. That's why they are called in polyar condition. In the xylem present in exarch condition. And the Casparian strips which found in the endodermis, they prevent or they make the cells impermeable to water. And as in dicot root, just remember the term. What is exarch, what is Casparian strip, pericycle, then what is the term used to describe the epidermis. So this is all about your anatomy of monocotyledonous root. So start practicing these diagrams and remember the terms which are being used to describe the arrangement, like a polyarch, diarch, then your exarch condition, then what is your vascular bundle, open type, closed type. Just focus on diagram and look beyond your environment, your surrounding, and see how these plants differ, monocots, dicots. Try to correlate your morphological aspects with the anatomical features. So it will bring curiosity in mind. So take care and we will meet your next class. Thank you.